Today we have to discuss the class 12th subject biology प्री बोर्ड एग्जामिनेशन. ठीक, चलिए शुरू करते हैं. सेक्शन ए. A farmer is advised to add culture of bacterium in the soil before sowing the crop. Name the bacterium in the culture. How is this bacterium useful to crops? 2 marks का है. Yes, the answer is Azotobacter. The Azospirillum. This bacterium converts the atmospheric nitrogen into the organic molecules that are used as fertilizers by plants. Next one. Explain HIV and AIDS and HIV, how they are different from each other. Or explain the role of the following providing the defense against the infection in the human body: B cells and interferons. The AIDS is acquired immunodeficiency syndrome. HIV is human immunodeficiency virus. AIDS is a condition of group of symptoms having been HIV infected individual. HIV causal organism of AIDS or any other differences. Second one is the produce antibodies in the response the pathogens antigens into the blood to fight with them. Glycoprotein which protects non-infected cells from further viral infections. Next one. Outline the structure of the drug is given below. Which group of drug does this represent? What are the modes of consumption of these drugs? Name the organ of the body which is affected by the consumption of these drugs. ठीक है, इस ड्रग का नाम बताना है. फर्स्ट ऑफ ऑल थर्ड का देखते हैं. दिस इज द कैनाबाइनोइड्स. बाय इनहेलेशन एंड इंजेक्शन. इनहेलेशन से लिया जाता है या फिर इंजेक्शन से लिया जाता है और ये हमारे कार्डियोवास्कुलर सिस्टम को अफेक्ट करता है. चलिए नेक्स्ट वन, क्वेश्चन नंबर 4. Define R in the population equation given by this one. dN upon dt is equals to Rn. How does the increase and decrease of the value of R affect the population size? R is intrinsic rate of natural increase. Population size increases with increase in R and decrease in R. Name the genus of baculovirus. Why are considered good biocontrol agents? Yes, answer fifth is nucleopolyhedrovirus. Don't harm plants, mammals, birds, fish and other target insects. Helpful in IPM. ठीक, कोई भी एक रीजन आपको बताना है. चलते हैं नेक्स्ट क्वेश्चन पर, दैट इज द सिक्स्थ वन. Why are some animals are rarely found in the polar regions? Explain. An orchid plant is growing on the branch of mango tree. How do you explain the interaction between the orchid and the mango tree? 6 का फर्स्ट पार्ट देखते हैं. The small animals like hummingbird have large surface area relative to their volume, so they tend to lose body heat very fast in cold. Second one, communalism. An orchid it benefited by the getting shelter from the mango tree, whereas the mango tree is neither harmer nor benefited. चलते हैं क्वेश्चन नंबर 7. Many diseases can be diagnosed by the observing the symptoms in the patient. In the list given below, observe the symptoms and write the name of disease. High fever, weakness, stomach pain, लॉस ऑफ एपेटाइट एंड कॉन्स्टिपेशन. इसमें कौन सा डिजीज़ है? इसमें है टाइफाइड. ठीक है, ज़्यादा बुखार है, कमजोरी आ रही है, स्टमक पेन है, लॉस ऑफ एपेटाइट एंड कॉन्स्टिपेशन, दैट इज टाइफाइड. नेक्स्ट वन, बी पार्ट. कॉन्स्टिपेशन, एब्डोमिनल पेन, क्रैम्प एंड ब्लड क्लॉट्स. पेट में दर्द हो रहा है या क्रैम्प्स आ रहे हैं, ब्लड क्लॉट्स हो रहा है, तो उस जो सिचुएशन में खून अगर जम रहा है तो वहाँ पर एमोबाइसिस है. नेक्स्ट वन, नसल कंजेशन, डिस्चार्ज, कफ, सोर थ्रोट एंड हेडेक. दिस इज द कॉमन कोल्ड एंड इन्फ्लुएंजा। ठीक है, आगे चलते हैं। नेम द सेल्स HIV gains entry in the after infecting the human body and explain the events that occur these cells. The macrophages help T cells correct sequence of the retrovirus cycle. Yes. Next one, question number 8. Why do the symptoms of malaria not appear immediately after the entry of sporozoites in the human body when bitten by the female anopheles? Name the host where the sexual and asexual reproduction of malaria parasites occur respectively. Give the scientific name of malaria parasite that causes the malignment of malaria in humans. Yes, the answer is the symptoms appear after the release of hemosin from the ruptured RBCs, female anopheles, and in humans, Plasmodium falciparum. Next one, question number 9. Suggest the figure of the vector PBR332 given below, which foreign DNA is ligated BMHI site for the tetracycline resistance gene. Mention the function of ROP. What will be the selectable marker for this recombinant plasmid and why? Which two scientists first artificially developed the cloning vector PBR322? The answer of the question 9 is ROP codes proteins involved in the replication of plasmid. Selectable marker ampicillin resistance gene. It will help the selection of transformants from the non-transformants after planting them on the ampicillin and containing the medium. The scientists are Bolivar and Rodriguez in 1977. Yes. 9th का और list and explain the 3 steps involved in the polymerase chain reaction. Alien species are highly invasive and are threat to the native species. Yes, बिल्कुल correct. Yes, the answer is yes, 9th का और जो है ओके 9th का और yes, denaturation as dsDNA is high temperature, annealing 2 sets of primers, extension primers of DNA by takes polymerase of deoxylitoids. ठीक है, ये 10th का ए पार्ट है जो मैंने एलियन स्पीसीज का पढ़ा. एलियन स्पीसीज बिकम इन्वेसिव, compete with the native species and cause of extinction of the indigenous species. ठीक है, कुछ बाहर के ऐसे पौधे आ जाते हैं जो वहाँ के नेटिव पौधों को जो है बिल्कुल विलुप्त कर देते हैं, उनकी जगह ले लेते हैं. ठीक है. नेक्स्ट वन, नेम एनी 2 कैटेगरीज इन विच वेरियस वेटलैंड्स टाइप्स आर ग्राउंडेड एज पर रामसर कन्वेंशन. रामसर कन्वेंशन वेटलैंड साइट्स का ये है, जहाँ पर वेटलैंड साइट्स होती हैं उसको जो है रामसर कन्वेंशन के अंतर्गत रखा जाता है. तो देखते हैं इसमें क्या है. यस, हाँ, एलियन स्पीसीज में कुछ एग्जांपल्स भी हैं. पार्थेनियम है, लेंटाना है, इकोर्निया है. ठीक है, पार्थेनियम घास जो होती है, जो आपने जनरली देखी होगी, वो एलियन स्पीसीज में आती है. ठीक है. रामसर कन्वेंशन 1971 इज इंटरनेशनल ट्रीटी फॉर द कंजर्वेशन ऑफ सस्टेनेबल यूटिलाइजेशन ऑफ वेटलैंड्स इन द फॉर्म ऑफ रामसर साइट्स which are wetlands are international importance. This convention work for the conservation of use of wetlands, recognition of fundamental ecological function of the wetlands and their cultural, economic, scientific, recreational value. These grouped as inland wetlands, marine and coastal wetlands and man-made wetlands. ठीक है, चलिए आगे चलते हैं. Study the graphical representation of the species area relationship given below and answer the questions that follow. ठीक है, you have to showing species and area relationship. YSCZ represents in the given graph. What will the range of the value of Z line if analysis of species are relationship among the very large area like entire continent? Answer of question number 11 is S is species richness, C is y-intercept, Z is slope of the line, A regression coefficient, and A is area. Z value will be in the range of 0.6 to 12. ठीक है. Yes. Next question. Explain any two factors less method to introducing rDNA into the competent host cell. How is DNA is isolated, is purified from the bacterial cell. A microinjection recombinant DNA directly injected into the nucleus of the animal cell using the micro needle tip of the diameter. Gene gun cells of bombarded velocity microparticles of gold and tungsten coated DNA implants. Bacterial cell treated with the lysozyme. RNA removed from the ribonuclease and proteins by protist following the DNA precipitation using the chilled ethanol. Yes. Next question. Question number 13. The two steps are involved in introducing the nematode resistant tobacco plant based on the RNA interface of this given one. Write the missing steps of this blank species A, C, D, F, G, H provided. ठीक है, देखते हैं. Why are scientific name of the parasite and part of the tobacco plant which infected? Isolation of nematode species gene, production of the sense of antisense of RNA in host, formation of the double stranded RNA dsRNA, silencing the species of mRNA and nematode. G is the host express the interfacing RNA. H is parasite nematode can't be survive in the transgenic host. Yes, second answer is meloidogene and incognita root. ठीक है, और पर चलते हैं. ठीक है, 13th का और. You have to go through this passage and this picture. Come to the question. Why is it called the proinsulin? Mention the chemical change that proinsulin undergoes to become a mature function insulin. Why is the insulin obtained from the other animal source not in use these days? What is the challenge in the production of the insulin using rDNA technique? Which transgenic species of the bacteria is being used in the production of human insulin commercially? The answers of 13 are is, it is called the proinsulin because it needs to be processed before it becomes the mature and fully functional hormones insulin. The C peptide is removed. The peptides A and B link the disulfide bridges. Insulin from the animal source cause the allergy and other immune reaction to the foreign protein. Hence, these are not used these days. The major challenge was getting two polypeptide chains assembled into the mature and the Escherichia coli. This was the question paper of class 12th subject biology. ठीक है, that's enough for this one. Thank you.